Okay, welcome to everyone joining us here. Today's talk is entitled From Muscle to Neuromuscular, Building 3D Human Functional NMJs with Mantarray. My name is Ariana, and I'll be helping guide us through this event today. This webinar is designed to be interactive, so if you'd all like to go ahead, if you haven't yet, and let us know in the chat where you're tuning in from, that would be a great start. Additionally, the webinar does include a Q&A session, so please feel free to submit those questions for our speakers anytime throughout the presentation. For those who are unfamiliar, you can find the Q&A and chat functions at the bottom of your Zoom screen. We will attend to the questions after the main presentation. Okay, now it is my pleasure to introduce our speakers for this event. Dr. Christos Michas and Dr. Jacob Fleming. To give a brief background, Dr. Michas is a product manager at Curi Bio, ensuring that the company's cutting-edge research translates into impactful products. He holds a PhD in biomedical engineering from Boston University while working with Professor Chris Chen, where he developed 3D microphysiological models of cardiac failure. Christos holds various patent and top-tier journal publications under his name and has a keen interest in the commercialization of innovative science. Dr. Fleming is a research scientist at Curi Bio, leading the Neuromuscular Junction project. His background is in skeletal muscle tissue engineering, with a focus on integration of additional cell types into engineered skeletal muscle tissues. Before Curi Bio, he graduated from the University of Oxford in biochemistry and received his PhD at Loughborough University while at Professor Mark Lewis's lab advancing engineered skeletal muscle models. He has published on a range of topics in the engineered skeletal muscle field, including muscle regeneration, maturation through mechanical stimulation, and models of the neuromuscular junction. Okay, without further ado, I'd like to hand over the presentation to our speakers. Christos, please begin whenever you are ready.
Absolutely. Thank you very much for the introduction, Ari. So hello, everyone. My name is Christos Michas, as Ari said, and I am a member of the product management team here at Curi Bio. And so today, myself and Jacob Fleming will be sharing with you our exciting news on modeling the neuromuscular junction in vitro. I will give you an overview of our established technology, and then Jacob Fleming will go into the details of the NMJ model he has developed. And so let me tell you a little bit about Curi Bio and what we do. Curi Bio focuses on the preclinical in vitro aspects of drug development and validation. The preclinical pipelines set the foundation for any drug program, and so it is not uncommon for problems to emerge when a drug is introduced to humans later. These problems are not identified in the preclinical phases, which to some extent defeats the purpose of having preclinical phases to begin with. So how can we make preclinical testing more predictive of what will ensue in humans so we can save money, time, and also tears down the line? And how can we do this in a scalable, reproducible, and automated manner that enables easy adoption in everyone's preclinical pipelines? And so at Curi Bio, we are aiming to do exactly that by integrating biology and engineering in products and services in a structure that we call the Curi engine. We provide cell resources that form the foundation of simple or complex in vitro models, and we enable the scalable fabrication of such models. We also provide specialized instrumentation, or what we call biosystems, that observe and perturb our models to extract data in a streamlined and standardizable manner. And finally, we provide software solutions based on advanced data processing and artificial intelligence to automatically convert the extracted data into meaningful interpretations to drive your clinical and preclinical decisions. Our products and services can be applied for drug discovery, for disease modeling, and for testing for safety, efficacy, and potency. And we are particularly proud to say that our technology is versatile, as it can flexibly adapt across many disciplines. And so in today's presentation, Jacob and I will demonstrate exactly how this same versatility can be employed to model a 3D neuromuscular junction in vitro. Before we continue into our technology, I would like to ask a little bit about you. So tell us, what part of preclinical research are you interested in? Please answer in the poll, select as many answers that are relevant to you, and we'll take a look at the results in a few seconds. All right, I see some results coming in already. Nice. All right, we see efficacy is a strong first, followed by disease modeling, and then therapeutic discovery. That's great to know. A few more seconds. All right, efficacy is where the people are at. That's great. Thank you very much, everyone. So for efficacy, how can we use this technology? And let me explain to you, first of all, what the core technology of Curi Bio is. So the core technology that Curi Bio leverages to make preclinical testing more predictive is our 3D tissue technology. So here in the pictures you're seeing on the left, that black strip is a 3D contractile muscle tissue. It is 8 millimeters in length, and it is one of the 24 tissues that one can make in Curi Bio standard consumable. We want to make the process of making these tissues as simple and easy as possible. And so the consumable has been optimized so you can create the tissues using basic cell culture skills, but also without the typical shortcomings of 3D tissues that are low throughput, low reproducibility, and slow procedures. We have addressed these shortcomings because we believe in the benefits of 3D tissues. And in a simplistic summary of those, the first one is physiological relevance. So now you have a 3D tissue that contains a unifying extracellular matrix with chemical and mechanical cues. And additionally, within that matrix, you have different cell types that interact with the matrix, but also with each other. Because the system is 3D, now you can access extended culture times. Typically, 3D cultures of contractile tissues last for months very easily, and so you can observe slow and potentially latent hidden effects in the tissues. And finally, you have access to function. That means you can monitor and record the contractions of the tissue, but you can also stimulate them and condition them with electromechanical conditioning for maturation and injury. So please keep these benefits in mind, because they will become particularly relevant as we're trying to build new models, such as the NMJ model. And so how do you use this technology? I have said again that we're trying to make this as simple as possible. How did we achieve this? So the Mantarray plate is split into two parts. There's at the bottom the maintenance plate and the tissue lid at the top. The maintenance plate is very similar to your standard 24-well grinder plate. It just holds the media. That's all there is to it. What matters, and that's the tissues, is in the tissue lid. So for each one of the wells in the plate, there are two posts in the tissue lid where the tissues are suspended from. And that means that if you want to move the tissues, all you have to do is grab the tissue lid. So to change media, for example, you grab another maintenance plate from us, you fill it up with your media, then you take the tissue lid and you move it over, and that was media change. It's so easy to maintain the tissues without destroying any of them. The same goes for casting. So we have the casting plate. All you have to do is place the tissue lid onto the casting plate, and by doing so, the two posts are going to end up in a shallow little well. On the side, you're going to mix with your pipette your cells with a hydrogel, and you're going to dispense that into the well of the casting plate. The cells are going to make the gel compact, and eventually you're going to get the nice little strip tissues that I showed you before. And this technology has been used time again by ourselves and our customers, to the point that we can brag that we have over 95% casting success rate. And since this technology has been validated so well, we have developed an entire series of services around it that span the entire spectrum of preclinical applications. If you contact us, we can help you identify iPSC sources for you, then edit them, differentiate them, expand them, place them in a proper 2D or 3D, like the one I showed you model, and then using the specialized instruments, which I will describe in a second, we can test your favorite gene therapy, your drugs, and collect data, and that data consolidated so you can have a report at the end to make your decisions. So in summary, we will have an entire study built, executed, and interpreted for you. And since we have done this so many times with my various customers, We all have also developed a series of well-validated products, so you can use them in your own labs. And so on the left, you see we offer commercialized cell lines of healthy and disease phenotypes for a variety of diseases. And the most notable example, I believe, is our wild type and Duchenne muscular dystrophy isogenic pair. Then we have proprietary media formulations that have been optimized to improve the longevity and performance of engineered tissues. And this goes back to the benefit of having extended culture times. We have a series of instruments in the middle and tissue culture consumables to run your own experiments fast and easy with very little technical expertise. And finally, we have a cloud analytics platform that's on the right to process your data and generate meaningful conclusions from your experiments fast and efficiently with a few clicks of a button. Speaking specifically about the instruments, we have 3 core instruments, and each one specializes to address specific needs and use cases. So we have the Mantarray, which is for contractility measurement and electrical stimulation, the Stingray, which specializes for stimulation, and then the Nautilite platform for electrophysiology. And all 3 of them are compatible with the foundational technology that is our 3D Mantarray tissues. And also they are compatible with the Pulse platform that is our cloud analytics platform that I mentioned just a few seconds ago. And so given this, let's talk about the Mantarray instrument. You see this on the left with and without a consumable. And within the consumable, you have the tissues that are contracting. And as they contract, they deflect, they move one of the 2 posts. And within that post, there is also a magnet. And so the Mantarray instrument is equipped with magnetic sensors right underneath each one of the wells. And the sensors track the magnet to convert it in real time, its motion, into force that the tissue is exerting. And this is done in a parallel manner for all 24 tissues at the same time. So this is a scalable solution. If you were to take a look at the complementary software interface that comes with Mantarray, then you would be able to see traces of all the forces that the tissues generate in the plate. And in fact, you can save this data and then upload it to the cloud to Pulse. And from there on, you can measure for each one of the contractions, not just how strong the contraction is, so the force amplitude, but also the time dynamics of it. So very fast, with a few clicks, you can fully characterize the contractility of your tissues. You can also modulate the contractility, and that is done with electrical stimulation using the stim lid or simulation lid longer. This is a consumable that you slide into the Mantarray plate. You connect it to the Mantarray instrument, and then you can assign to each one of the wells a different simulation protocol. And this is again done very easily with the software interface of Mantarray. For example, in this case, we are assigning for the leftmost column of the plate, just those 4 wells, a protocol that you're seeing at the bottom that consists of 4 sub-protocols, each one with different colors. And each one of them has different stimulation parameters, which could be the stimulation current, the amplitude of the current, whether it's monophasic, biphasic, the stimulation frequency, etc. And by doing this, you have the option of driving different contractile states. So for a skeletal muscle here, we show a twitch contraction, so just small twitches, or drive continuous contraction in the form of a tetanus with 10, 20, or 30 hertz. And this is really good for characterizing the contractility of tissues. But then this same approach is very useful because you can validate different disease phenotypes. And in fact, it might even save you from long, costly animal studies. In the example that I'm showing over here, at the very top, we have taken our wild type and Duchenne muscular dystrophy isogenic pair, and we have made 3D skeletal muscle tissues out of that. And we have driven them into tetanus. And you're clearly seeing that the DMD line is incapable of keeping up with the wild type force production in the tetanic states. And this is an observation that has been made time and again in animals. You see that at the bottom. And so this very simple example is a clear proof that our 3D technology and our cell lines can be used to replicate, complement, and potentially even replace animal studies, which are expensive, inefficient, and in some geographies will be phased out eventually. So this is with the Mantarray system. Then we have the Stingray, the second instrument in the list. And so this is specialized for stimulation. It is the same convenient design as Mantarray, same user-friendly software interface, same stimulation capabilities, but it is at a fraction of a price. So you have multiple and you can have scalable stimulation for maturation, for injury, or for running any other study that you see necessary. And finally, we have the Nautilite platform. This is our electrophysiology instrument. It utilizes fluorescent probing to measure calcium flux and membrane voltage. It is, of course, compatible with the 3D tissue technology that I have described, but it is also compatible with 2D technology, and it can go up to 384 wells, so the throughput is substantially higher. The instrument records optically the sample fluorescence, and then using the Pulse Online platform, the cloud analytics platform, you can calculate the fluorescence time dynamics, just like with force before, but also you can calculate other relevant metrics, such as conduction velocity, action potential, duration, and triangulation. And even though the instrument has the substantial high throughput, it still manages to preserve very high signal to noise ratio and high temporal resolution. So you can clearly identify ephes events and analyze their time dynamics. So for example, on the left here, you're seeing 3D tissues that are placed in the Nautilus instrument, but are being stimulated externally by a Stingray instrument. And so as the tissues contract, we have sufficient temporal resolution to drive them into tetanus and notice even the small details, the contractile details at the top of the tetanus. And so given this description of our technology, I hope you can understand that Curi Bio provides sufficient and excellent cell resources and instrumentation so you can introduce 3D muscle studies in your lab. And that means cardiac, which I haven't really explained in this presentation, but also skeletal muscle. But as cell resources improve, so as the iPSC technology improves, and for example, we have new lines, new stem cell derived lines like neuronal ones, a question to be asked is whether this technology is sufficiently versatile to adopt these new opportunities. And it is. For example, we have managed to build the NMJ system by combining neuronal and skeletal muscle iPSC cells. And Jacob will go into the details of this model in a second. So as we are making this transition to that part of the presentation, I would like to ask you, have you used NMJ models in the past? And if you have, please tell us if you met any significant limitation that restricted the use of them. If you have not used any, then please feel free not to answer to the second question. All right, some results are coming in. Seems that most of our attendees are new to this, which is excellent because Jacob has really exciting results for you. Data extraction has been a problem for one of our friends. A couple more seconds. All right, it seems the consensus is that we have a lot of new people regarding NMJ, and so Jacob, you can take it from here.
That's great. Thanks, Christos, and nice that we've got plenty of people new to this because hopefully you'll find it informative and exciting if this is your first interaction with an NMJ system. Christos illustrated there really quite mature technology ecosystem. We're really pleased with the Mantarray system. When I began working with 3D engineered tissues, these were very bespoke, difficult to work with, and not scalable or reliable in people's hands that were not experienced. That just isn't the case now. We can hand these systems off to people and expect success. And so we're now asking, can we go further? Can we make the biology more complex? Can we make it more meaningful in terms of its relationship to real in vivo biology? And in vivo, skeletal muscle does not contract in response to electrical stimulation from carbon electrodes as it does in our system as it currently stands. Instead, contraction is evoked through acetylcholine release into the neuromuscular synapse, which drives an action potential on the skeletal muscle membrane and causes contraction through calcium release. And the neuromuscular junction is one of these structures that's truly three-dimensional. It has a complex ECM that is patterned on these invaginations, for example, and seals the synapse space from the intracellular space. And so it's a really good opportunity to leverage the strengths of 3D models and ensure that the model we produce is meaningful. Perhaps modeling something like the neuromuscular in 2D, where you have these short experimental timelines and the inability to create these complex structures, doesn't give you the opportunity to model real biology. And the neuromuscular junction lies at the center of everyone's lives. It's important in locomotion. It drives the respiratory system and therefore is fundamental for survival. And so diseases that affect it, or small molecule inhibitors, such as neurotoxins, have devastating effects on individuals' lives. So the ability to model it, replace animal models, and improve the predictability of these preclinical testing platforms has the potential to really change people's lives who have diseases such as ALS, SMA, and myasthenia gravis. And we're not necessarily the first people to think of this. People have been using two skeletal muscle model systems for some time. And so the extension to the neuromuscular system has been seen before. There's a couple of papers at the top left there, one from Penny Gilbert's lab in Toronto and one from Mark Lewis's lab in the UK in Loughborough, that are very similar in some ways to our own. They're the approaches that I would point you to as the most similar that existed before. But there's been a range. There's organoid systems here, for example, all the way from stem cells. There's microfluidic systems. Some people may have seen Roger Cam's microfluidic system, which is 3D as well. And there's also been attempts to compartmentalize larger tissues too. And this is just a selection of a broader and quite significant literature base, but they've all come unstuck somewhere where it comes to commercializing or handing them off, or even reliably reproducing them within their own labs and publishing again and again. And these really comes down, I think, to a few things. The first, perhaps, is the scalability of it. Can these systems support at least, say, 24 tissues, which can be cast reliably and respond every time when you try to examine the neuromuscular junction? Can they be manufactured reproducibly? And that's something that often microfluidic systems come unstuck on. Can we really make these things at the scale that people want to use them at? And how reproducible is the neuron response? If I set up 10 or 100 of these cultures, am I getting 90% to 95% showing innovation and neuromuscular junction formation? Or, and I think this has really been the case to date in the literature, are we seeing 20 or 30%? So those are some of the challenges the literature has faced and previous attempts have faced. And so we wanted to take the ethos of the Mantarray system, where we see 95% success in terms of functional tissues, and create an NMJ model that was the same. So how have we gone about this? We've taken the Mantarray casting trench that Christos described, where we cast the cells and the hydrogel in between the two posts, and we've embossed upon the base of it a neuromuscular, a spheroid creating geometry that promotes the creation of neurospheres when we seed neurons as single cells into this trench. Those neurons will compact into neurospheres, mature in the bottom of that trench, and we can then remove the media which we've been maintaining them in, apply on top an acellular hydrogel. And before that acellular hydrogel is set, we can take the tissue-containing lid that Christos described, and just like that media change, we can move it over into the casting trenches and place it into that hydrogel that's still liquid, adhering the neurospheres to the outside of this mature skeletal muscle tissue. And so what we've now created is a co-culture. And this picture here with the red arrows illustrates that. We have the skeletal muscle on the inside and the neurospheres adhered to the outside. This is fantastic. We brought the two cell types together, but we now have two challenges. how do we activate our motor neuron action potentials independently of the muscle so that we can show innervation and neuronal directed function? And the second is, how can we maintain skeletal muscle function and neuronal function, so axonal extension and neuromuscular junction creation, in the same media, because these tissues are now multicellular and within one well. So the first challenge, neuron-specific activation. Our approach has been the introduction of a blue light sensitive channel rhodopsin into these iPSC-derived motor neurons. The introduction of that channel has not affected the differentiation. We still see fantastic motor neuron differentiation. They create these spheroids and we get excellent axonal extension. These images were taken 24 hours after placing down on plastic, and we get extension of over 1 millimeter a day with these motor neurons. They express CHAT and islet-1, classic markers of the motor neuron lineage. And so we're really happy that these are healthy, mature motor neurons ready to create neuromuscular junctions. In terms of the blue light activated action potential, this trace in the top right shows some MEA data, microelectrode array data. The black arrows at the bottom are blue light incidents, and you can see the blue light incidents aligning with these peaks in the electrophysiological activity. So those blue light channels can be activated and drive axon action potentials. And because the underlying muscle does not contain these, the exposure to blue light allows specific activation of the motor neurons. On the co-culture point, muscle function perhaps is our key measure here. Can we maintain our skeletal muscle within this co-culture media and still see function across time? And the answer is yes. We have a comparison here between a muscle-only condition and a condition which contains neurons and muscle in the co-culture media. Force is extremely similar and not different at all, I don't think. Time to peak, or the time from 10% of the waveform to the very top, remains the same as well between those two conditions. And actually, the only place we see a difference is in this kinetic measurement of half relaxation time. So that's the time from the top of the peak to halfway down the curve. We see slight decreases in the co-culture relaxation times. And that's actually consistent with some of the literature that to date that's shown improved skeletal muscle maturity. For example, increased force and increased half relaxation times are good examples of increased muscle maturity. So this may be an indication that the neurons are beginning to affect the underlying skeletal muscle biology in a positive manner. One of the first things that people look for is spontaneous activity of co-cultures versus the monoculture. The reason for this is that skeletal muscle alone is not spontaneously active. It will sit at rest unless a stimulation is applied. But motor neurons have got a relatively high amount of spontaneous activity. And so once the motor neurons connect to the skeletal muscle, the spontaneous activity trickles down through the synapse and causes this kind of sporadic, spontaneous contraction. It's a really useful measure. It's easy to see. It gives you a clear indication that you have a neuromuscular junction, but it's quite difficult to analyze. If I asked you, what are the width of those peaks, or what's the height of those peaks? The answer is not straightforward. We have multiple populations, perhaps. We perhaps have two populations in terms of height there, some standout tall ones, and then these smaller ones down the bottom. And then we apply a drug or perhaps a disease phenotype. How does this change? How do we quantify that? And those questions can become really challenging, to the point actually where the system becomes unusable. So this is where we have to think about this specific activation of the motor neurons directing a response, a functional response to the muscle. And so this is the blue light activation of those motor neurons, and you see this trace here. These blue bars indicate when the blue light has been shined on, and we see this fantastic correlation between blue light and functional response of the underlying muscle. This is force generation that we're looking at, and we can do this at 5, 2, and 1 second intervals. Using that stimulation system that Christos has described, we have really high levels of control over the type of stimulation we apply. And we can run those peaks through our Pulse 3D, our online analysis tools, and get absolute force. we can get the relative force of the blue light response to the electrically stimulated response. And this allows us to estimate how innervated the tissues are. So say a tissue is producing 20% of the force with blue light that it does when electrically evoked, producing its maximal force. We can say perhaps 1 in 5 of the myotubes within that tissue are innervated and are responding to the neuronal signal. So it gives us some way to estimate the proportion of innervation. And then percentage capture is how often is the response to blue light seen. Well, actually, it's seen every time in this system. At the lower incidence of blue light, the lower stimulation rates. And that small drop from 100% at 1 second is actually due to the beginning of fused tetanus. So instead of seeing 3 individual peaks in that 1 second pulse train, we see perhaps 1 or 2 of those fusing together to produce something that looks like tetanus. So it's not necessarily the absence of response, it's that there's already a response ongoing. And that's really important to have that high fidelity in response, because there are diseases out there like ALS, for example, where the loss of response fidelity is a disease phenotype. And we really need to see high fidelity in our responses. One of the things that NMJ research has done a lot of, and for good reason, is to image and analyze the morphology of the NMJ. It produces fantastic images, the NMJ. It's one of those biological structures that's exquisitely organized. And there's good reasons to look at it, because that organization builds across time during development. And the pre and post-synaptic markers are key indicators of disease state and developmental state. And so in order to access disease modeling, we really wanted to be able to show that we could image, even at a basic level, the NMJs that we have. And so in this image, we've stained for myosin heavy chain for myotubes, TUJ1, which is an axonal marker, and will mark up all of the neurons in the culture. And we've used Bungaroo toxin to identify acetylcholine receptor clusters. And what we're looking for is points where all 3 of those stains come together, and that is an NMJ. And those points are highlighted, or some of them, some examples are highlighted here with the white arrows, but actually there's a large number of these. And that's a key point. We want to see large numbers of NMJs, because that's going to make it much easier when you're trying to image one to find them within your cross-section, stain up, and get meaningful data with enough replicates to make meaningful interpretations. And then the other thing that this image shows is a clear separation between the neuronal population and the muscle population. This bottom right shows the full cross-section. You can see the muscle on the inside and the neurons on the outside, as we'd expect, having adhered to the outside. But it's suggested that we're not seeing just a bulk electrophysiological response where perhaps an axonal body pressed against a myotube is causing a functional washover. Instead, that gap suggests this probably is a specific interaction. But to make absolutely sure, we wanted to block that response with a neurotoxin that acts only at the NMJ. We've chosen Botox for 2 reasons, actually. The first reason is that Botox has a complex biological mechanism. It's a 4-step activation mechanism to cause its effect: uptake, enzymatic cleavage and activation within the endosome, release into the cytoplasm, and cleavage of the snare complex on the presynaptic membrane. This relies upon the assembly of a great deal of important apparatus at the presynaptic membrane, and is a good indication that some level of maturity in the NMJ has been reached. And the second reason is that Botox is a biological product. It's created for medical use and cosmetic use. Through growth of the bacteria and isolation of that toxin, and the multiple step processes mean that those manufacturing systems have inconsistent potency results. And this means that every batch of Botox needs to be tested for potency before it can be released. And it's currently used through a mouse lethality assay And we're looking to replace that with some ex vivo assay using this system that removes the need for animal testing in the potency testing of Botox. We're also excited about the potency testing because there are other examples of this, especially in gene therapy, where a lot of genetic diseases in skeletal muscle are heading. These gene therapies are similar to Botox. They require complex multi-stage manufacturing processes. And in order to batch and lot release these types of therapies, we're going to need potency assays. And this work's funded through a grant through the NIH, and we're working closely with the FDA as well on this to try and get a potency assay approved. And it's going, I think, to prove extremely useful going forward in other potency assays for complex biologics that we might want to develop. So those are the two reasons we've chosen Botox, perhaps rather than one of the more obvious neurotoxins that might be out there. Here's the data before we apply the Botox on the left. The control and the Botox group look virtually identical, but 4 hours later, the Botox treated group, the signal in response to blue light has been almost completely eliminated. And we can quantify this. This is a group of N of 4, and we see a good time-dependent response to that Botox treatment. And that's to be expected in line with that multi-stage mechanism. Botox takes a little while to become active, cause its effect. And you may say, perhaps that Botox has been toxic to the muscle and we've reduced function that way. Well, we can go in and do broad field stimulation with the electrodes after we've treated with Botox, and the response of the skeletal muscle alone, driven by electrical stim, is identical between the control and the Botox group. So that's not what's happened. And we could also say perhaps that the neurons have been killed by the Botox, but actually this blue line here on the top right graph drops following the Botox. So this is the blue light evoked response now. It stays low for 2 weeks, but then we begin to see reinnervation. So there's clearly a population of neurons that are still viable and can reinnervate the tissue. So we haven't wholesale destroyed the neuronal population either. And taken together with the data on the previous slide, I think that shows a really clear, specific interaction of the neurons and the muscle as an NMJ and not some non-specific interaction, which could have occurred. And the final piece of data that I'd just like to draw your attention to is the stability of the control on this top graph. We see stable NMJ function for over 3 weeks in this model, and that's really important for diseases like ALS, for example, which are progressive diseases where you see slow, steady changes. And so you need stable models and able to be able to study them. So how does this fit in the Mantarray ecosystem? We're going to need some way to stimulate these tissues with blue light. And what we've done is we've built an optical stimulation lid using laser diodes, and each well has a laser diode associated with it, a focusing lens, and this puts a very specific spot of blue light on the tissue. And you can control this through the same stimulation software that Christos showed. This allows high intensity, high precision stimulation with temporal linking to the recordings, allowing you to align stimulation response. So just to close, that we have this model now, we can evoke neuronally directed muscle contraction, we can visualize the NMJ. What next? Well, I touched on the Botox potency assay. That's an exciting application, and hopefully will push us into an interesting new world where we can really demonstrate the replacement of animal models with an ex vivo model. We're looking to begin some disease modeling. We have collaborators at the University of Washington who are working with us on some of the Botox work as well, who are excited to begin some ALS modeling with this system. And we're also interested to see if neuronal derivative maturation occurs in the underlying skeletal muscle, because we know in diseases of the neuromuscular system where neurons poorly innervate or do not innervate the skeletal muscle system, it doesn't mature properly. We see fiber type switching, we see atrophy. And so we understand that there's a key role for the neurons in skeletal muscle health and disease as well. And so that's it. And it would be great to have any questions that you've got. I think it looks like we do have one in the Q&A, and Ariana will be back, I think, to go through those and ask us.
No, absolutely. Thank you so much, Jacob, for the part of the presentation. So you're absolutely right. We are in the Q&A portion of our webinar. And so if you do have any questions, please submit them in using the Q&A option. And we do have the first question already from Joseph. So great presentation. Thank you very much, Joseph. My question, how do you control for the number of cells, for example, myoblasts and or myotubes in each 3D construct?
Yes, so that's to do with the seeding of the myoblasts into the casting trench at the beginning. There are two key aspects here. We thaw our myoblasts directly from the liquid nitrogen, and we don't expand them on plastic at all. They go straight into the casting wells with the hydrogel and directly into a differentiation media. And so we're hoping by compressing that growth phase as much as possible, we keep better control of cell number. So the number that you put into this hydrogel is as close as possible to the number that then go on to form myotubes. So hopefully that answers your question. That's how we try and reduce variability as much as possible on that.
Awesome. Second question from a different Joseph this time. So a remarkable number of NMJs looked apparent in the cross-section images you show. Have you established whether each fiber is innervated by a single motor neuron, as what happens in vivo?
We haven't. And so that's a fantastic question. That, I think, touches on that complex biology around synapse pruning and all of that interaction that goes on to stabilize the NMJ and the competitive interactions between motor neurons. I think right now that's probably beyond the scope of what we're looking to do, but I do think this model would be perfect for it. And so if you have an interest in that type of work, I really think this platform would be a good model for it.
Thank you. Let's see. Another question by Alex. So is the optogenetic stimulation lead available in the Curi Bio catalog? Well, I can take this one. So right now, the technology that Jacob just presented, so this is internal technology. We have not made it commercial just yet. And so I would say specifically for the optogenetic stimulation lid, this is compatible with the Mantarray stimulation platform. So it's the same as replacing it with the stimlet, the electrical stimlet. So it should be very easy to adapt. If you are interested, and this is also a broader comment, if you're interested in working with the platform, the system that Jacob has developed, then please do reach out to us to contact@curibio.com. You see that email at the bottom. So we can get in touch and see if we can collaborate or if you can get to test your system in your own lab. Another question, do you need to differentiate your motor neurons in situ, or can you use cryopreserved progenitors?
We can use cryopreserved progenitors, much like how I just explained with the direct thaw into the hydrogel for the muscle tissues. We can take the motor neuron progenitors directly from the liquid nitrogen, place them into that casting trench geometry I described, and they'll create neurospheres right there without an expansion phase. So you don't need to do your whole differentiation in that consumable.
Another question from an anonymous attendee, is the NMJ system compatible with mini tissues? Jacob, do you want to take that one?
Yes, I can take that. That's so I think this must be one of our beta testers on one of our new products, the Mantarray mini tissues. So they are a smaller version of the standard Mantarray tissue. It reduces the cell number required, and so takes some pressure off cell production. And so we're hoping that'll be a step forward for people. But actually, all of the data I've shown you has been based around the mini system here. So yes, they are compatible with the mini system. And I think that smaller tissue lends itself really to innovation because there's less distance for the motor neurons to work through.
Thank you. Another question, again by Joseph. Any plans to integrate other cell type structures into Mantarray? For example, myotendinous junctions, vasculature, satellite cell regeneration models. I can answer part of that, and Jacob, you can also pitch in if you'd like. So this exact presentation is an example where we are trying to expand beyond our original core muscle program. And so, yes, we do have plans to expand. If you have ideas on how we can do this as well, we would happily get in touch with you so we can discuss further. But the system as we have it right now is amenable to changes, so we can introduce different tissue types. And Jacob's the introduction of neuronal tissue neurospheres is a prime example of how we can do that.
And yeah, and it's almost like you were looking over my shoulder during my PhD and postdoc. I have worked with vascular cells, myotendinous junctions, and I published a couple of papers on regeneration models in these tissues. They do contain PAX7 positive resting cells, which appear to be within a basement membrane. And so when it comes to satellite cells and regeneration, I think we can say they're already there. And myotendinous junction and vasculature, much like the NMJ, require some additional work. And like Christos said, we might be interested. We'd have to sort of see how it all fits.
A question from Manmeet Raval. Hey, Manmeet. Have you characterized the fiber isotype composition of the muscle tissues in the NMJ system?
We have not yet, and it touches on that kind of muscle maturity and fiber type switching and all that exciting pieces of biology that happen post innovation. I'd love to do it, perhaps right now, beyond the scope of what we'd be looking to do. So if you're interested, get in touch and perhaps we can figure something out. But I would love to see, is the innovation changing the skeletal muscle biology underneath?
One more question. How are the co-culture medium conditions different compared to the standard Mantarray tissues alone?
So the base media is based on our skeletal muscle media that Christos mentioned. That's a proprietary media, but we do supply it, plus a series of neuronal factors, which are also proprietary, but they are all part of that differentiation process and are designed to support axonal extension and neuron health. And so it's a combination of our muscle media and some extra neuronal factors.
Awesome. Well, I think this is the last question we'll be answering. So we reached the end of the Q&A portion. And if you had a great question, but you didn't get to ask it or we didn't answer it, please do reach out to us at the email you're seeing on the screen, contact@curibio.com. It was a great pleasure presenting to you. Thank you, Jacob, for helping with this. And please enjoy the rest of your day and feel free to reach out to us. Thank you very much for joining.
Thank you, everyone.